put it another way. This is the evolution of developmental biology in the absence of an organism that develops. So Dictyostelium are organisms that are single-celled. They have single-cell amoebae, like amoeba, but plural, amoebae. And they do some really cool things, though, that mean that this is one of the most frequently studied developmental biology organisms, despite the fact that it doesn't actually develop. And the reason for that is because, evolutionarily speaking, a lot of developmental biologists consider Dictyostelium to be sort of the closest ancestor to multicellular organisms. So it's a comparison between what happens when you get multiple cells, and therefore development can occur. Which I guess raises a really important question that we've never really talked about. We did briefly. What's the definition of development? What are the essential components of a system that has development?
It's able to, I guess, replicate from one into something, or propagate and stuff.
Okay, so organisms propagate. Organisms always propagate, right? That's one of the general principles of life, is replication. So what's different about, and we're going to see this a little bit in Dictyostelium, what's different about them than what we might consider higher organisms, although that's kind of a biased term. So at the first class, I think, or maybe the second class, I mentioned that we weren't going to be studying any single-celled organisms. I was wrong, because they don't do development. So on one hand, organisms that do development are multicellular. If they're not multicellular, they don't develop. But there's a definition or a concept that underlies why multicellularity is a requirement, but it isn't the sole definition of development.
I guess it has to have some differentiation between cells.
That's development. Different cells in one body that do different things. So you have to have differentiated cell types. That's what we're going to see in Dictyostelium. This is one of the oldest, I guess, organisms that evolved the ability for one body, kind of, we'll see what I mean by body, can have different cell types. So we're going to talk quickly about the evolution of multicellularity, and then we'll talk about the issues with having developmental biology, which is that some cells, if they start to differentiate, could become cheater cells. We're going to work a little bit on the concept of what a cheater cell is and why that's only possible after you get development started. And then I'll tell you about the biology of Dictyostelium. There have been lots of different independent evolutions of multicellularity from different parts of the tree of life over time. And, for example, animals, plants, and fungi. And we're going to be talking about the latter today. So what's difficult about multicellularity? If you were God and you were setting up the rules of life and you were creating organisms, what would be difficult about creating a multicellular organism?
I don't think like they're communicating between the same language. So do I have to develop different ways they communicate between each other to create some different cell types?
Right. So there has to be, if there are different types of cells, then the cells necessarily, or maybe necessarily, I don't know, we could argue about that, have to be able to communicate with each other. Set up different regions of the body. Now, I'm an eye cell, you shouldn't be an eye cell because you're next to me in the wrong direction. So cells need to be able to communicate with each other in a multicellular organism. Otherwise you've got chaos. Every cell deciding on its own what type of tissue to become, and then you've got brain cells next to muscle cells, next to bone cell. Yeah, that's one of the main challenges to the evolution of multicellularity. A single-celled organism by itself has DNA, it makes all the proteins it needs to do all of its functions. As soon as you go to, say, a two-celled organism, any multicellularity, then those cells have the same DNA in them, the exact same DNA, same chromosome sequences, same chromosomes, but if there's going to be differentiation, then genetic changes have to occur to those chromosomes to allow different cells to become differentiated, different types of differentiated cells. So there's a lot of genetic change that's involved in gene regulation change. That is, one cell needs to be able to turn, as we've talked about a lot this semester, some cells need to be able to turn off the expression of some genes while turning on the expression of other genes. So it's complicated. So there are a number of challenges to overcome, and it's one of those things where people just sort of sit back and scratch their heads and think, how did that ever actually happen? Much like the transition we're going to talk about later this semester of aquatic animals to terrestrial animals. People that believe in creation, I believe in creation, but I also believe in genetics. So people that exclusively believe in creation and not genetics and evolution make the argument, well, how did we ever get transition from you're breathing oxygen dissolved in water with gills and no limbs, and then all of a sudden there were these terrestrial, how did that happen in one step? It wasn't one step, it was multiple steps, but all of those steps were necessary to go from life underwater to life on land. And similar claims in terms of conceptual difficulty can be made here. How is it possible that any series of a lineage of organisms could possibly have done all of this to evolve from unicellular organisms to multicellular? Multicellular organisms must have just been created. So difficulties, but we'll see at least a little bit how difficulties can be overcome. One of the nice things about developmental biology and about multicellularity is you get cooperation. It lets me do something like walk, because my cells are cooperating together, and I can do a lot more than I could if I was a single-celled organism. There's another form of cooperation that is important to understand, and this gets a little bit beyond typical evolutionary biology and talking about cooperation between individuals, which is a little bit more like sociality, and I'll leave it at that. So there are some evolutionary concepts that basically revolve around a concept called kin selection that we might be able to talk about a little bit more today. But the idea, I'll just leave it at that. So you've got cooperation between cells to form an organism, but you can also have different types of cooperation where different individuals cooperate. Like if you work together with a lab partner to complete an exercise, you're both contributing to a process that you both think will benefit you. Ooh, this is a good analogy. Maybe I should bring up cheating now. What's this sort of cheating individual in the lab group working on a project in a lab course scenario? There are cooperators and there are cheaters. There's some organisms in a group that can be cooperators, but then what's the cheating individual?
Somebody who doesn't do anything.
The one person that doesn't do any of the work and still gets the credit for it, and everybody's mad at them, but they're still part of the group and they still get points. That is a cheater organism. They pretend like they're part of the group, but they don't actually contribute to it, and they benefit from it. That's not cooperation, by the way. That's cheating. Cooperators and cheaters are two different cell types. They're two different people types in different situations. They're also two different cell types. So here's an example of, sort of, an example of kin selection. Cooperation among individuals. A lot of different insects, especially Hymenoptera, tend to be cooperative. We've got ant colonies, we've got bee colonies that all work sort of collectively to the common good. And the one difference really is that the queens tend to live longer and all the other workers tend to have really short lives. But this is eusociality, cooperation of organisms to fulfill sort of a greater purpose. So there's some issues to multicellularity, but what are some benefits to being multicellular? That is, benefits to having diverse cell types. What can we do when we're multicellular that we can't do when we're unicellular? I know it's kind of hard to imagine because you were unicellular once. I know each and every one of you was unicellular once, a long time ago.
Sexual reproduction.
So there's a benefit to sexual reproduction in form of, for evolutionary biology, that's diversity. So the creation of diversity. It's easier to be genetically diverse when you have combinations of chromosomes from different individuals coming together into organisms.
Maybe one cell knows how to do fix one of the other cell's problem. I guess they
So you-
complement each other.
Yeah, they can cooperate.
Mechanisms that the other one doesn't have.
Okay, so specialization. So you can do a lot more different types of things as a multicellular organism than you can as a unicellular organism. That's why I've got a liver that can detoxify both alcohol and benzopyrenes. I've got different cell types that produce different enzymes that detoxify different components, different chemicals. Other thoughts?
Longer lifespan.
So longer lifespan for multiple reasons. One might come back to the fact that there are then some cell types, or there are some tools that cells can use when they're multicellular to extend their lifespan. Complexity.
If like one type of cell stopped functioning, it wouldn't just die.
That's right. You're not just a single cell, and if you die, the whole organism's dead. Maybe you've got some stem cells and you can replace that cell, or you're a planarian and you can regenerate, except for if you're human. Sigh. So there's some costs to multicellularity. There's some potential benefits. So a long-standing debate has been sort of how do we explain how multicellularity evolved in the first place? Because there's always a cost and a benefit to doing something. So there's a cost to being multicellular. There's also potentially benefits. So understanding them both is relevant. So here's a cost. This is the main cost that people think about when they talk about costs of multicellularity or of sexual reproduction, speaking of sex. It's the same issue, actually. If you're a unisexual, how do most unicellular organisms reproduce? Think bacteria.
Asexual reproduction.
What does that one bacterial cell do to make offspring?
Fission something.
Right, so usually a bacterial cell will divide in half, make 2 cells. Then those 2 cells will each divide, and you get 4 cells, and 8 cells, and 16 cells. This cell division is growth. It's reproduction. So plants and animals don't do this. Most of the cells in a multicellular organism will not be the ones that pass the genome on to the next generation. That is a cost, evolutionarily speaking, because the organism is investing a lot of energy into building cells that have a function, and they're important, but that don't actually directly lead to the transmission of genes to the next generation. Hence this slide. If the plant's sole purpose was reproduction, then it would make all of its tissue gametes, fruits, basically. But then there would be no plant. It would just be all fruit, and the plant would die, and then the genes wouldn't get transmitted to the next generation. So there's, again, this concept of a trade-off. The organism has to put enough effort into building the body that will ensure the transmission of a few cells to the next generation to propagate the lineage, the genealogy. So there's this balance between putting too much investment into making gametes and not enough investment into making gametes as opposed to the soma, the somatic structures, everything that's not reproductive tissue. I probably should have put this slide first. But if you were an evolutionary biologist, what's the purpose of life? We already covered it. Reproduction. If you don't reproduce, non-reproduction, therefore, equals death. Because from the evolutionary biology perspective, we're thinking about over time scales, we're talking about over generations of cells or generations of individuals. if you're a cell that doesn't pass your chromosomes on to future beings, then your lineage is extinct. It's basically death. It's the equivalent of death. So when a cell stops dividing, it's still alive in that it's part of your body and it's still doing biochemical functions inside it, but it's never going to make any more kids. So to a cell biologist, that's death. That cell is dead. It's still functioning, but it stopped reproducing. It stopped dividing and making daughters.
Why do some scientists think the viruses are not like living things? By themselves, they can't reproduce.
Right. That's a tangential but relevant point that, viruses, whether or not viruses are considered alive, is hotly debated because they can't survive on their own. They're more like parasites. But then parasites are alive, but most parasites have a form where they live outside of the organism that they parasitize. So what is a virus? A virus also doesn't do a bunch of other things that living things, well, not a bunch. So all living things also metabolize. They have to take in energy, and then they excrete. They have waste products, not necessarily feces, but some sort of waste product. And do viruses do that? Because viruses hijack the host cell to make the components of a virus. So, So if you're not reproducing anymore, that's evolutionary death. So here is the point that's critical for Dictyostelium, for these slime molds especially, is how do you transition from, this is theoretical, this is something that they're going to test in this paper, but it's sort of a theoretical, philosophical question that we could all sit around and stroke our goatees about, or not, depending on whether or not you have a goatee, is if you're going to evolve from unicellularity to multicellularity, you've got to convince, without brains, because cells don't have brains, but you've got to convince some of the cells in the organism to die. You have to convince the body cells, whatever the multicellular organism is going to have some cells that are the germ cells, that are the ones that are involved in reproduction, that will pass their genes on to the next generation. But all the other cells in the body of the organism, whatever multicellular organism it is that evolves, therefore those cells are giving up their opportunity to reproduce by being part of the body. That's a cost to those cells that give up the right to reproduce. So that's the essence of this question, this conundrum of how does multicellularity evolve given this trade-off. All cells, to an extent, especially unicellular cells, are selfish. They each benefit from reproducing as much as they can. Bacteria, for example. Bacteria that reproduces, that divides faster, grows faster than its other bacteria in its environment. So its progeny get more access, earlier access to whatever resources are in the environment. So they win. So there must be some benefit and some cost to being multicellular. Because if it was just being multicellular for the basis of being multicellular, then that would not be selected for. It's not advantageous just to be multicellular unless you can do some cool things. Like reproduce, have developed tissues, distinct tissue types. What's another benefit of being, going back to a slide from a couple moments ago, what's another benefit of being multicellular? In terms of senses, if you're a bacterium and you're in a region of the world that's devoid of nutrients, let's say you're a bacterium that can move, so you've got a flagellum, let's say, and you're in liquid, and there's some nutrient over there on that side of the room. What does the bacterium do?
Usually it has like chemokine receptors that steer that way sometimes.
Okay, so there's bacteria have senses. They can tell sort of where things are located in their environment. But bacteria are pretty small. And so what's important for, what's the value of any sort of sense? Let's especially think of smell. what do you have to do if you're an organism and you can smell to either go towards something good or go away from something bad that you can smell? So let's say I was standing here and I smelled something horrible, like formaldehyde. Like imagine this is the lab that's on the other side of the building, the embryology lab, and you don't like the smell of formaldehyde. So if I'm here and I smell formaldehyde, what do I do to get away from it? Which direction do I move? Wait, but how do I know which is the opposite direction? Because I'm a bacterium, I'm just sitting here. Something's bad somewhere in my environment. So how do I know which direction to go? I want to go away from the bad smell. All I know right now is I'm smelling something that's bad. Where's it coming from? What do I have to do? How do you figure out where the bad smell in the room is coming from?
Concentration?
So what do I have to do to figure that out? As a unicellular bacterium,
Move around?
I have to move around. So if I move this, it's like, well, it's that one game. What's that game? Hot and cold, right? I have to go this direction. And if if I start smelling more, if I start sensing more of that compound, then you know I'm going to go this direction. So that's another benefit of multicellularity is not only are we mobile, maybe more so than unicellular organisms, but we have body axes, like head to toe, and if I had noses down here too, I wouldn't even have to move. I would know that my toes are smelling less formaldehyde than my head. So I would know immediately, without moving, I could tell which direction there's a chemical gradient because I've got sensors across my body. This is directly relevant to what Dictyostelium does, so don't worry, I don't have toes in my nose and toes in my nose. So for multicellularity, you do have to have cells be able to convince and talk to other cells. There has to be cell-cell communication for some cells to say, hey, I'm the germline. You guys go off and do building the body stuff. We're special. We're going to pass on the chromosomes and you all are going to do other stuff. And so I'm just showing a couple of examples that are classical examples. This does not have any direct relevance to the paper. They're just examples that in eukaryotes, especially developing eukaryotes, there is a process of gastrulation. This is a ball of cells after fertilization, so several hundred cells with an empty cavity in the middle. And there's a particular stage of development where cells that are on the outside migrate towards don't have to remember this, the dorsal blastopore lip. There is a hole that goes inside of this cavity. And the cells will migrate and then go inside the gastrula, which is what this is called, this whole ball of cells. And when cells migrate over this thing called the dorsal blastopore lip, there are cells there that put out a particular signal. And when cells that are crawling along the surface and then involuting, when they pass over those cells are giving instructions to the cells that are migrating over top of them. They're giving out, like we talked about last week, there are different signaling pathways, JAK, STAT, hedgehog, and Wnt. Those cells use those sorts of pathways to inform cells as they're moving across, you're going to be this type of cell, you're going to be that type of cell. So this is something that happens in development of most organisms, is cells send signals to other cells to tell them what to be. The same is true for eye development. You don't have to know the details, although this involves fibroblast growth factor signaling, which we've seen in the past, where tissue inside the organism sends signals to the ectoderm to tell certain spots on the ectoderm you're going to start becoming an eye cell. This is the evolution of cell-cell signaling is really common. And we've seen these before. These are the cell pathways, or the signaling pathways that many of those cells use. So hedgehog signaling involves one of these freely diffusible factors, we've talked about this in past weeks, that can be sent over short distances across multiple cells. That is not just cell-cell connections, but these are diffusible signals. Can be used to tell regions of organisms what types of cells to be. So for development, we have to have cell-cell signaling, and we have to worry about cheaters. And this cheating concept is what this paper that we're reading is all about. And there's a theory. Can I help?
Just gonna pick up something from.
Please do. Go for it. So cheaters can spread, this is just a theory, cheaters can spread in asexual populations. So what this means is cheaters are mutants, basically. A cheater type of cell is different than a cooperator cell, and they're genetically different. So the theory is that you could get, if you were a cooperating cell in a multicellular organism, you were doing things that was benefiting the greater good, you could get a mutation that would cause you to cheat. You would be acting like you were participating in the group activity, but you really weren't. So in theory, you get benefits from being part of a big body because you're defended from the elements or from predators or whatever. And so the idea here is that in asexually reproducing populations, if a mutation arises, let's say you've got a population of cells, and one cell in red evolves a mutation that allows cheating. If this population of cells is only asexually reproducing, that means that each cell just divides and produces another cell, then if all of these cells divide, then that red cell will divide too. That is, there's no cost to being a cheater. Everyone's replicating as quickly as everybody else. So at this point, this red mutation is not spreading, but it's not going down in frequency either. It's not being selected against. Cheaters aren't being removed from the group by the group members. But there are opportunities for those cheaters to spread because by chance, some of these individuals that are asexually reproducing that are cooperators might get killed or not be in environments where they have enough resources or something like that. So by chance, over time, those cheating mutations, DNA mutations in individual organisms can spread. They can become a bigger part of the population in terms of frequency of cell types. And of course, some of these might die randomly too.
So the cheater population, they only benefit from the cooperation of the others, right?
Right.
So is it like a commensalistic relationship, or what do you think?
So there is a problem with being a cheater, and I'm glad you brought this up, and this is something that the authors of this paper definitely discuss, which is what happens if you're a cheater in a multicellular organism. What happens if all of the cooperator cells die? Or what happens if there are enough cheater cells that the whole organism can't function because there are too many cheaters that aren't actually doing their organismal jobs or their cellular jobs? What happens to the organism? It could die. If there's too much cheating, then the group doesn't turn in a product that gets any points. The group dies. Your grade dies, something like that. So there are a lot of these similar theories in evolutionary biology about what should happen if cheating arises. If cheating evolves, then that's probably detrimental to the organism. Is there a threshold at which there's so much cheating that the organism dies? How do organisms that I guess sustain cheaters, how do they survive? Is there an optimal level? If you were a cheater cell, these are all sorts of questions that evolutionary biologists ask. If you were a cheater cell, how successful do you want to be at cheating? Because if you cheat too much, you kill your organism that you're cheating from. But then you're also a cheater, so you want to pass your genome on to as many offspring as possible. So there must be a balance there. So how do cheater cells find that balance? In other words, again, is there an optimal level of cheating that would evolve? Cheating, again, remember, we haven't gone into details, cheating is mutation. These are DNA mutations that cause organisms to act slightly differently, to act selfishly, to not do as much for the organism, but they still benefit from being part of the organism. So maybe there are different types of mutations that are just cheating enough to allow an organism to survive, but to allow the cheaters to continue to live in the organism and propagate. It's really weird. Theoretical evolution.
They have some sort of sense to their environment. There's not enough resources. You gotta let the others.
It's hard to think about how this happens in sort of evolutionary terms. You have to think about it on a multi-organismal level. Every time an organism, a multicellular organism, has a cell that evolves cheating, that organism is tested against natural selection. Is that organism more or less fit than the other organisms around it? So in this paper we're going to read, what the authors do is they do experimental evolution. That is, they look at, they give, in this case, Dictyostelium, which we'll talk about in just a minute, literally thousands of independent opportunities to evolve cheating, and then they want to study what are the effects of cheating. Every time it evolves, do you have strong cheaters? Do you have weak cheaters? The theory is that if you study the evolution of cheating enough times, you would be able to identify, is there a type of mutation or an optimal level of cheating that seems to be best fit, that allows the organism to survive but still lets cheaters evolve. Okay, so that's quite enough of the theory, I think, although there would be a lot more to discuss if we really went into detail. But to appreciate what the paper is doing, I think that gives you at least some hooks in your brain to hang some of the introductory material from the paper on. So the main reason this paper got into Nature, I will leave for, oh wait, I'm presenting this paper, so I guess I'm not taking it away from anybody but myself to tell you now. The reason this paper got into, I think it was in Science, it was either Science or Nature, pretty sure it was Science, is nobody's been able to do this research before to study the evolution of cheating, because when cheating evolves, the organism dies, and then you have no organism to study. Remember, the reason that cheaters kill the organism is because once cheating evolves, then cheating spreads in that organism from generation to generation. There are more and more cheaters, and eventually there are not enough cells doing their job.
Does that always happen?
Well, that's a good question. Does it always happen? And the answer is, I don't know. And at least they didn't know at the time they did this, because of this problem, is that you couldn't study cheating, because when cheating evolved, then you weren't left with any organisms to study. So there was no idea of how frequently cheating evolved, how long it could stick around in terms of numbers of generations before that lineage might go extinct. What happens to a lineage once cheating evolved? Can anti-cheaters evolve, cells that would punish the cheating cells to revert from cheating back into an organism that was entirely cooperator cells? I know this is all very abstract. Cooperators, cheaters, cells interacting with cells.
So how, I'm very like confused with these and cancer. Would that be like what you're talking about?
So is cancer a good example of a cheater cell? I think it is. I think I might go a step further and say that cancer cells are cheater plus cells. They are doing things to their own advantage to propagate themselves at the detriment of the host. So in that sense, I think they're a classical form of cheater cell. Interesting, thanks. That might help me think about this paper better too, because sometimes it is hard to come up with a good example of what a cheating system would look like. I'll have to think more about whether it fits all of the definitions of a cheating system, but that's a good example.
In cancer, the thing that's causing the cancer usually messes up with the cell cycle, so the cells keep dividing, and that leads to tumors, and that leads to cancer.
Right.
But I don't think these cheater cells are doing that. They just don't do anything, and
But
they eventually cause the cell to die.
right. So cheaters, from what I've read about cheater cells, which is not necessarily not by any means anywhere close to the number of cheater cell papers in the field of evolution, would argue, I think that most of those papers would argue that the cheater cell has a replicative advantage, because the concept of cheating in evolution is to pass your genome on more frequently than other cells. Whether or not that strictly fits a cancer definition, I'm not sure, but definitely worth thinking about. Another cells, and of course, cells don't have brains, so cells don't know that they're cheaters. But a cancer, a mutation that causes tumorigenesis, that is a mutation that ignores cell cycle regulation, so that a cell keeps dividing over and over and over again without regard for what's going on in the body, sounds like a classic form of cheating, which is, yeah, I'm trying to propagate myself as much as I can, as quickly as I can, even though I'm not the cell that's supposed to be doing the passing on of the genetic material to future generations. I like it. Thanks for the input. Ponderous. Okay, so Dictyostelium is a type of slime mold. I'm showing some examples of different species of slime molds here, just to highlight how I don't know, they're beautiful. They're also slime molds. And so slime molds, Dictyostelium discoideum is the species of slime mold that these authors are talking about, but slime molds in general have this really cool life cycle, which is explained on the next slide or 2 slides from now. These are the multicellular versions of slime molds. So slime molds can exist, not just can, exist as unicellular amoebas, just one cell crawling around looking for nutrients. But in certain circumstances, which I will tell you about in a moment, they will group together and form individuals that are multiple amoebas, but these organisms, these bodies that these individual amoebas form, kind of like transformers, has unique properties that unicellular amoebas can't do or can't have on their own. Oh, and you may have seen this recently. Over the last week, I've been seeing a bunch of news articles about this slime mold, Physarum polycephalum, the many-headed slime. So my wife said this to me, actually, because she read this and said it had 720 sexes. This one species has 720 different sexes. And so that's why she sent it to me, because I used to study sex chromosomes and sex determination. She said 720 sexes. But anyway, this slime mold, no, this isn't that slime mold that does weird things to snails. But there's a new exhibit at a botanical garden that's specifically growing this slime mold to demonstrate its really cool properties. And so apparently I don't remember where, I think it was in France or maybe Europe, or England, I mean.
Zoo.
Which one?
Brooklyn Zoo.
Is it Brooklyn? Okay. I'm glad thanks. Oh, I was probably thinking of Kew, which is in England, but that's not where this is. So yeah, it could be in Brooklyn. They've actually made a new exhibit with this slime mold growing on a tree. It's a huge slime mold, and it has these really cool properties, like so it grows in thin streams towards food, usually, and so if you cut the slime mold, then it can heal itself, even though it's a bunch of independently acting individual organisms, but all together, they have these cool properties. This paper was cited by one of these, this just came out, this paper suggests, although this is going to be controversial, I think, that slime molds can habituate. That is, if you expose slime molds, individual amoebas that group together that form a long string of connected, sort of, amoebas into a big multicellular-ish organism, can remember if they've been exposed to something that's noxious in the past and then avoid that sort of chemical in the future. So it seems that what these authors are arguing is that this is a unicellular organism that has a multicellular form that it can remember, despite the fact that it doesn't have a nervous system. So I might pass this on to Dr. Lent, and then you can go take his neurobiology class if you haven't already, and ask him all sorts of interesting questions about whether or not it takes a nervous system to remember. Oh, let's see, what does this say? This is the abstract. Right. So here we demonstrate habituation in a non-neural organism, an organism without a nervous system. Showed that this slime mold learned to ignore quinine or caffeine. That's crazy. So do slime molds have memories despite not having brains? So here we are, studying another species of slime mold, this Dictyostelium discoideum. So here's how it works. Single-cell amoebas are happy, they roam around on their own. When the organisms start to sense that food supplies are running low, nutrient availability is diminishing, then one cell, whichever cell is maybe most individually aware that resources are dwindling, will start sending out a cyclic AMP signal, a small molecule that one cell will secrete. And so that cell that secretes it here produces then a gradient. Right? The concentration of this chemical is closest to the organism that's emitting this signal, and the concentration of this chemical is lowest at the outskirts. So this gives a gradient that other amoebas can follow. They're attracted to this signal. So once one organism starts emitting this signal, then a bunch of other amoebas will join it. They move towards that, and that's this streaming. Lots of individual amoebas moving towards one spot. And then those cells accumulate and turn into a form that's called a slug. So this is one group of individual amoebas that moves around together. It looks like a slug, like a classic slug outdoors. More in the Pacific Northwest than in California, but I've had some slugs from time to time in my yard. And so one of the benefits of the slug is that it has polarity. So it's like the nose on the head and the nose on the toes. A slug benefits from being multicellular because it's got length, so it can tell whether it's moving toward or away from different chemicals, because the cells, the amoeba that are part of the head of the slug, can sense, if they're closer to a high concentration of something, that slug knows that it should move in that direction because the tail senses a lower concentration of that same signal. So it's a three-dimensional organism, so now it has polarity, and it can sense where to go. So the slug will actually move around. You can grow these on petri dishes. We use this all the time in Biology 155, developmental biology. Love playing with these things. You put some slime molds on a plate. Once the slime molds eat all the food on the plate, then they all form slugs, and you can see them crawling around, not in real time. If you take a time lapse, you can see them crawling around on the plate. And then at some point, they will decide that it's time to disperse even farther than just the petri dish or wherever they're growing in nature. And so that slug will turn into a fruiting body. And this is the tricky part. This is the multicellularity part, because in the fruiting body, the spores, which are the gametes, they're the cells that then go on and try to colonize new regions of the world that maybe have more food. Those are at the tip of this stalk. So there's a base, there's a stalk, and then there's cells at the tip of the fruiting body, and those are the spores. And presumably the reason for the fruiting body is that the slime mold wants to get up high in the air so that some of the spores might stick onto some passing insects or something. For whatever the story is, it evolved to do this. So slime molds do this. And that was what you saw in a couple slides ago with the different colored slime molds. They had all grown fruiting bodies to put spores up somewhere in the air or on the tips of the stalks. So this is the problem then, is that only a handful of the cells are going to become spores. These were all formerly original independent amoebas, each living on their own. But now some cells are going to have to tell other cells, you are going to sacrifice your lives to become the stalk. And we're going to go up the stalk and spread our genes.
So is are they genetically related, where that would be?
So, one of the important concepts here is kin selection, which is the concept that cells that are genetically identical or individuals that are genetically similar to each other might be willing to sacrifice themselves to help pass on gene or genealogies that are very similar to theirs. Because if you can't have kids of your own, maybe the next best thing is to help your sister or your brother take care of their kids, because their genes are pretty closely related to you, their family. So if you can't have your own kids, maybe it's beneficial to help your close relatives succeed. So that's the concept of kin selection. And that is possibly what's going on here. That's one way to argue for some cells to convince some cells to become somatic cells and others to be gametes. So you can actually watch this happen. It's really cool. Again, it's time lapse. Over the course of a day or so, you can watch this happen. Oh, I already said that stuff. There you go. And you can do things with these organisms like you can with planaria. You can cut them in half and see how they act. They reform two. You can make one slug form two slugs. And one of the really interesting things, I don't know if I kept this in the slides or took it out, is that this slug, not only is it multicellular and it's acting like it's one organism, it's held together by a cellulose sheath. So the cells that are on the outside of the organism produce cellulose to hold all the cells together. So there is some differentiation of cells and what the different cells do in this organism. But people also discovered over decades, this has been known for a long time, they could, say, label certain cells, and they discovered that there are, in this slug, this is already a I don't know what word I want to use. There are already tissues, basically. Aside from the fact that the cells on the outside of the slug produce cellulose, the cells that are in the tip or the head of the slug already are fated to a particular outcome. And that is, the tip cells are the ones that may become the spores. So even in the slug phase, you can already predict which cells are going to be the ones that are going to be the gametes and which ones are the ones that are going to be the stalk. It's like the slug has already decided which cells are going to pass on their genes to the next generation and which ones are going to become somatic tissues. And with that knowledge then, the fact that in a slug there are certain regions that if you were a cheater cell and you wanted to be the ones that were going to go up the stalk and be the spores, there are some types of mutations that might be cheating mutations. So in this case, if you were an individual amoeba and you wanted to make sure you got to the head of the slug so that you could be one of the spores, if you had a mutation that let you crawl a little bit faster, maybe you could make it up to the front of the slug where you would be placed up in as one of the fruiting bodies. Another type of cheating mutation might be you produce some sort of chemical that prevents other amoebas near you from responding to cyclic AMP, for example. So you prevent other amoebas from traveling as quickly, like you say, nobody pay attention to the signal that you're getting. I'm going to go that way, but you guys stay here. Right. So that was this is a question from long ago, was how do the cells that form the fruiting body of the stalk actually know how to get there? And there's been a lot of work done, which we do not need to get into details of, what genes and what signaling pathways tell different cell types what to do. But the point is that this is already known in Dictyostelium. There's been a lot of work done on figuring how these cell-cell signaling pathways, notch, hedgehog, those sorts of pathways, are used to tell different types in the context of a slug what to do. So here's that idea that the pre-stalk cells actually go to the tip of the mound. So if you were a cheater, you'd want to actually not go to the top of the mound because that's where the stalk's going to form. You'd want to be down in the base somewhere and get carried up the stalk. So there are lots of ways in theory, for amoebas to develop cheating mutations. So these are the sorts of experiments that people did to tell where the cells are that do different things. Was they would label, for example, certain types of cells with GFP. And so they could see where in the slug different cell types are and where they wind up once the stalk forms. And they could figure out what areas of the slug form different parts of the fruiting body, the base, the stalk, and the tip with the spores. So all this is done before. And the reason that finally we can get some answers to the questions of what happens when cheating forms is these authors reason anyway, we'll see if it's true. The authors reason that this might be a great system for studying cheating because these organisms can live both as individuals and as multicellular organisms. So you can't kill a multicellular organism, I didn't tell you this yet, but you can't kill this multicellular organism because if you break the cellulose sheath of the slug and just distribute all the amoebas, they go back to amoeba life. So you can see, and this is how the authors are going to do this, you can see the effects of cheating by mixing, and this is the experimental evolution part. I'll get back to how they do it in a minute. So they're going to grow different populations of Dictyostelium for multiple generations in replicates. They take one genotype, close relatives of each other, and they make lots of different independent cultures of that. And then they want to know, does cheating ever evolve in any particular lineage? So you grow, I don't remember how many, you'll see it in the paper, hundreds or thousands of different replicates of starting with a single amoeba, letting it grow, and then seeing, does cheating ever evolve, and if so, how frequently? So they're going to use two different experimental approaches, two different types of experimental evolution. The low relatedness approach is they take a lot of amoebas and they grow them all together, and because it's a large population, if any one individual evolved a mutation that was a cheating mutation, they would probably be selected against because all of their other colleagues, the other family members, are cooperators. So you're not likely to see much, you're not likely to see much competition from the family members if cheating evolves. So, okay, so here we go. So the authors made 24 different replicate lines. They start with 100,000, I think, amoebas in each of these populations. They grow them in large numbers for 31 generations, 24 times. And because it's a large population, you're not likely to see many mutations, but if the mutations occur, then there's a huge benefit if you are a cheater, because there's lots of suckers around. There's a lot of cooperating cells around. So if you happen to be an organism that involves cheating, you benefit from it. And that's why this says selection is high. That is, there's a huge selective advantage to being a cheater in this case. The other type of experiment they did was basically the opposite. Instead of a large population, they use a small population. And instead of no population bottleneck, which basically just means you feed the population as much food as the population needs, in this case, they grow a very small number of organisms, and every generation they take one, or every few generations, they take one amoeba to start a new population, to continue that lineage. And this is a situation where natural selection doesn't act because it's artificial selection. The experimenters are picking one individual from a huge population to be the founder of the next population. So that's the opposite of natural selection. There's no opportunity for cheaters to be disfavored, in other words, in small populations, because it doesn't matter if you're a cheater. If you happen to be the one individual that gets picked by the experimenters to found the next generation, that's great. So they don't expect cheating to evolve there, because what happens if you're the cheater and you get picked to be the one representative in your population that founds the next generation? If you're a cheater and you just produce more cheaters, that's death. There are no cooperators anymore. So in this side, with small population sizes and bottlenecks, that is, you let the population grow to a large number of individuals and then you pick one and let it start the next generation, that's called bottlenecking. With small populations and bottlenecking, you're likely, if cheating evolves, to pick one and then that lineage would go extinct. So you don't expect cheating to evolve there. And you do in the opposite situation where you have large population sizes and no bottlenecks. You just feed, let them grow as much as they want. That's ideal conditions for cooperation, and therefore that gives opportunity for cheaters to benefit, because this is a cooperating like situation. Makes my brain hurt. So don't feel badly if your brain hurts too. So that's, last slide, that's what they're going to do. So what they do is they grow equal numbers of organisms from any two of these replicates. So the the small population size, the large population size. One's likely to have mutations that cause cheating, one's unlikely. So you fluorescently label, you take, say, 300 of the non-cheating, maybe non-cheaters, and 300 of the ones from the population you think might evolve cheating. You fluorescently label these 300, you mix them together with the unlabeled ones from the other population. So you know, you can look and you can see which ones are the cheating cells, theoretically, maybe cheating cells. The test then is, you let those organisms grow until they starve and form fruiting bodies, and then you take the cells that become the fruiting body, and you ask, is there still an equal mixture of the two populations, the unlabeled and the labeled? If the labeled population has cheaters, relative to the other population you've mixed them together with, what's likely to happen when you look at the cells that are in the fruiting body? The cheaters are more likely to be in the fruiting body if they're cheaters. So you're looking for non-50-50 ratios. You're looking for a fruiting body that's all cheater cells, that's all from these one type of experimentally evolved lines compared to their ancestors. So the important last point, the important thing to note from here is that when you have slugs, and I said you could cut them and separate them and see how they operate individually as two halves of a slug. If you dissociate a slug, you can mix the slugs together and then they'll form new slugs. So that's basically what they're doing here, is they mix equal numbers of amoeba from different types of organisms, different types of treatments rather, and they'll form slugs that are a mixture of both types of cells. So if there was a cheater, you could have a half cheater cell, half non-cheater cell slug, and that's when you'd be able to see these effects. So if you had a slug, it could be made up of an equal number of, let's say that's red cheater cells, blue non-cheater cells. So when this forms a fruiting body, if cheating has evolved during the experimental evolution of the lines, then you'd expect most of the non-cheater cells to be in the stalk, maybe a few up in the fruiting body. You'd expect most of the cheater cells to be the ones that have gained a mutation that will help them preferentially pass on into the next generation. When the expectation would be there'd be an even mixture of blue and red cells throughout the whole fruiting body and stalk in the absence of cheating. So we'll see what they find in a month or so. Any questions? Other than why did I tell you about this now, since we're not going to talk about this until 3 weeks from now? Who's talking next week?
Oh, I am. I'm talking.
Awesome. What's our topic next week?
It's feathers and scales.
Ooh, nice. Yeah, are feathers and scales related? We'll see. So I will see you. Remember, don't come on Wednesday. I'm sorry I have to be at a meeting. I will see you next Monday.